to run a workshop on extracting DNA. But when I started to do this, we were in communication about what we really want to do. And it occurred to me what we're really doing here is not extracting it so much as preserving it. Because the extraction goes on in a lab later and is sort of beyond us. And that's a good thing. It's like something we don't need to deal with. But what we do need to deal with is we need to make sure that the specimens preserved, the DNA in the specimens are preserved, and that we can then extract it easily to get a sequence later. So that's what I want to talk about, and I'll be quick because it turns out it's easy. So the goals of DNA extraction are basically to stabilize and then to purify the DNA. And the type of extraction that you're going to do is going to depend on the type of material that it is, animal, plant, fungal, whether it's root tips or whether it's mushrooms or maybe whether it's polypores, could change that. And also how good the DNA has to be that you're getting out. So if you've got, if you're going to sequence an entire genome, you've got to get quality DNA out. But that's not what we're doing, right? What we want usually from these specimens is one or maybe two or three sequences of particular genes in a relatively small little piece. So as a result, the DNA doesn't have to be very good from this. It can be pretty crappy, actually, and it doesn't have to be that pure, especially if we're going for sort of an abundant gene, because you can dilute out the impurities and you still have enough DNA there to get the sequence. So we can settle for pretty crappy stuff. So that's good. And that's just what the next one says, so I can move on to it. I just want to give you a little background on the properties of DNA in terms of the stability. And the take-home message is that it's a highly stable molecule that really you have to work to destroy DNA. And so that works in our favor. It can get nicked or broken mechanically by shearing, but as I was mentioning earlier, we can settle for smaller pieces, so that's not a big issue for our needs. It can get damaged by some chemicals like phenolics and tannins and stuff, and this actually can be a problem, especially on plant DNA, that sometimes plant stuff complexes with the DNA makes it difficult to use thereafter. With most fungi, this isn't a huge problem. And over the long term, oxygen and water damage it, but that is the long term. We're talking, decades before the kind of damage that this would inflict on you.
But does it matter? I mean, once you've done the extraction and you get a sequence, does it matter what happens? I mean, do you guys actually preserve the DNA in the long term in kind of a herbarium setting?
There's been a lot of talk about this, about if you're going to extract from a specimen, maybe you should preserve this DNA long term, because down the line maybe somebody else will want to sequence something else. And the answer is no, we've not done that. And by and large, it's not necessary, I don't think, because you can always go back to the specimen if you preserved it in the ways that you just heard about. You can always go back to that specimen and get more DNA back out. Or your rotten card, which we're getting to. So overall, DNA is pretty tough stuff. Now, in the short term, there's only very few things that will break down DNA. One is strong acids, like much stronger than we're going to experience unless you're like pouring battery acid on the things. Very high temperatures, things that would burn it, basically. Like boiling it is gravy. It kills everything else, but the DNA is preserved. So very high temperature, basically. And then specific enzymes called nucleases are very efficient at breaking down DNA, and those are the ones I want to underline. That is, when DNA breaks down in a specimen in a short time, that's the cause, is that nucleases are doing it. And the fungi themselves and any kind of biological material has nucleases in it. And they're usually compartmentalized in the living cell in a way that they're separate from the DNA. But when that cell dies, those compartments break down, the nucleases are released, and the DNA will get chewed up very quickly by the nucleases. So the main thing that you're doing when you're extracting DNA or stabilizing it in the context of a specimen is you're making sure those nucleases do not have any time at all to work on the DNA. Because if they do, then there will be no DNA. That's the main culprit. So how do you get rid of nucleases? You can do this by denaturing or activating them. High temperature, not so high that it destroys the DNA, but high enough to kill the nucleases, because nucleases are proteins, right? So anything that denatures a protein will denature these. You can use strong detergents, and that's very typical in extractions. There's some kind of like SDS or CTAB you hear about. These are just detergents, and they basically denature proteins, so they get rid of nucleases. You can all nucleases need divalent cations. They need, and usually magnesium, and so putting in chelating chemistry gets rid of those. So a lot of times buffers that you use for DNA have a strong EDTA content because it's just pulling out the cations, and when that happens, the nucleases get inactivated. And then some kind of strong non-polar organics can also do this. So phenol and chloroform are sometimes used in extractions too. Now this is all stuff you don't really need to know. And I'm filling time because I could go right to the point that it's very simple.
So is there like an optimum, or does it matter what temperature we dry specimens at? Or is that basically better? Is that a concern for the microscopy later rather than the DNA?
I think no. I think as long as they dry quickly, the temperature probably does not matter.
Okay.
What you want to do is drive the water off as fast as you can. So high temperature can do that, but it has other adverse properties for the microscopy later. These modern food dryers that we have that that have a forced air are really great. They do a great job of drying the specimens quickly, and that will preserve it. So that's the other thing, to inactivate nucleases, just dry them out, keep them dry. So, punchline, what's the fastest, cheapest way to preserve DNA in a mushroom specimen? Dry them out, dry the specimen, keep it dry. So all the things that you just heard about vouchering and stuff, if you just do that, it will preserve the DNA in the specimen. And if you're in kind of an area where there's a high humidity, like maybe you live in Vancouver, and you're getting a lot of humidity there all the time. Well, sometimes specimens can be hygroscopic and rehydrate and so on. When you dry them, when they're dry, put them in a Ziploc bag and zip it up. It'll stay dry. It'll cut it off from that external moisture, and that'll keep for a decade easily.
Not if it's really humid.
Even in a Ziploc bag?
I lost stuff at home because it was at home where it was all humid. Yeah, it's a Ziploc bag, so.
What about a sealer jar?
What about silicon gel?
Yeah.
You can add a desiccant to it too. But I have to say, the Bay Area is pretty humid in the winter, and
This
most
is like
specimens
5
last
years or
forever.
something, I don't know.
Quite a long time. And some of the specimens have been in the herbarium for decades. You can still get DNA out of them and sequence small regions. Now, as they get really old, it's more of a pain to do, and the pieces you get are small, so it's more difficult. But it's usually still there. So I think that when you are making these collections at home, for example, you find that cool thing, and you're not on a foray like this, just dry it out. Keep it dry, and it'll be fine. And we can get the DNA out of it later. Okay, but for large numbers of samples from a foray, then we want to simplify these methods and archive the DNA separately from the specimen, so that we can take the DNA and send it off to a lab that's going to finish the extraction and sequence it, and we can take the specimen and send it off to a herbarium and catalog it and put it in a box and label it. And we want those two connected still. But dealing with them separately will speed up the process. So that comes to these things. And I've got them sitting around here somewhere. Just had them out. Here they are. They're these little cards. And the way these things look is they've got an inner part of them. So, and there's 4 little spots on there. And what happens is that you just put a little bit of material on here, like a little piece of a gill, typically. And then on the outside of the card, you're going to put a label for what that is, as the collection number, so we keep it together. And then you just hit it with a hammer and air dry it. And it's supposed to be good for decades like that.
So are you putting 4 samples on one, or just
So you can
one?
put 4 samples on one.
From the same thing or from different things?
From different things. So you have, 4 collection numbers here, and then you have 4 little pieces inside.
How much material do you need to do DNA analysis then? Would you just like a pinhead or?
Yeah, you don't need very much. You need about as much as you would use for, probably a little bit less than you would need for a microscopic examination of the very same specimen.
So fingernail clipping.
Yeah, I think on the handout I said about 5 millimeters by 5 millimeters. That would be oodles. I could.
There's chemicals in the paper.
And so exactly how this works is not fully disclosed, because they're selling these things to us for $4 or $4.50 a card. And I'm guessing all it is some kind of a silicon-based filter impregnated with EDTA and a detergent. So I don't, but that's my guess as to what they're doing. What they say in their literature is that it denatures the nucleases and it binds the DNA. And then you get back to the lab and there's a longer process to extract it, but there's actually a shorter way to do it, which I'll mention briefly. Where do you find these things? Amazon. Yeah, you can. I was just looking for an image of it on the web, and I typed in, FTA card, Walkman card, image.
There's only 2 left.
At that price. Yeah, you can see that the 2 new ones you can get for less than their regular price. So here's their box of 100. It'd be $459, and apparently somebody's got a couple new boxes that they're willing to sell for less. We really should experiment and find out if we can make these things for next to nothing, because I think they're selling us something for $4.50 that probably cost them a nickel to make. But even at $4.50, it comes down to just over $1 a sample to preserve it, right? So it's not that bad. And then down the line, if you look at the handout, I give some estimates of what it's going to cost beyond that. I would guess for most labs it's going to be somewhere between another $5 and $10 to produce a sequence out of this in the end. But it's relatively quick. And you can go to YouTube and see how this is done. Bryn Denninger put this up on YouTube. Let's see if we can get it to play here. I know I can get this to work. You don't have the sound. Okay.
Is your sound down?
My sound down. Ah!
That was the problem yesterday.
There you go. All right.
When collecting new specimens for DNA laboratory, the first thing you want to make sure you do is get a good photo of it in the field. Later
could
on,
supplement that and
you
say a
sample
good-
DNA from
a
the
good
specimens
collection.
using these Robbin FDA Plant Saver cards.
Oh, could you sit down?
These cards have 4 sample areas and a laminated flap that folds over the sample area. Take a clean, fresh piece of reproductive tissue.
Look at how small this
Here I
is.
pour off some tubes of fluid and place that tissue in the middle of one of the sample areas. You want to be careful when you place the tissue in the sample area so you don't damage the surface of the card. Then you want to fold the flap over and use a hammer to smash the tissue. Next, check and make sure that you properly smash the tissue by observing the back of the card and looking for a stain on the back. This stain indicates that the cellular contents of the tissue have been broken open and have soaked through the fibers in the card. There you can see the stain. You want to remove excess tissue that accumulates on the flap as well as on the surface of the sample area. And be careful when you do this again to make sure you're not damaging the fibers of the card. It's important that you label the sample right away and then allow the cards to air dry. Or if you're in a humid environment, you can place the cards in a Ziploc baggie with desiccant. And that's it.
Tough, huh? We could have cut to the chase here. You could have saved several minutes here on my presentation. But very simple, and we have used this. We had a much smaller foray on the initial collections at Yosemite Park, and we had sort of an assembly line of photographing and then hammering these things, and they extracted very well. And the other thing is that the extractions from this point on are actually very, very easy. And Denninger also published something on that. There's a teaming it up with another kit, the extractant amp kit from Sigma. Allows you to punch these things out, put them in a microtiter dish where you have 96 wells, so you can do 96 of these at once. You add buffer, you cook it for 10 minutes, and that's it. You just add, after that, you add a second buffer to it, and it sits overnight in the refrigerator, and then it's ready to be used the next day. So it's like no time at all to extract once these cards are made. And so that's the real advantage of them, is that if at a foray like this you take the time to bang out your specimens on the cards, somebody in a lab will need very little time to extract everything you've done. So here today I brought something just over 60 cards, I think, or something like that. So that would give us more than 200 specimens that we could do. That would be 2 microtiter dishes plus. So it would maybe take 20 minutes in a lab to do. So it can be pretty fast.
The rationale for removing the excess tissue is that crucial? And what's the rationale?
I'm not sure what the rationale on that is. One I guess, is that it could it could dry up and fall over on other different areas of the card. So it might be partly just a cross-contamination issue. It also, if you have a lot there, it could decay. The part that's going to be preserved is going to be the part that's interacting with the fibers of this card. So the stuff that's up above there is not going to be preserved, and you might as well remove it. Any questions on this?
How well does it work with polypore stuff? Stuff?
So I did try polypores, and he said it worked okay, but not as well. And that was our experience too. That especially if it's a fairly dry polypore, it's probably not worth the time to do this. You want things with a fairly juicy cellular contents.
Right. You don't do it for like wet deep crusts. Things where there's not much material.
No.
Sorry, what was your comment? I didn't hear you.
So some of the fungi that people study, like Tom and other people, are really obscure little crusty fungi on their logs. There's not much tissue there, so
They're not really suited to this. And the polypores he was mentioning are also not as well suited, although they can work, they just don't work as well usually. The juicier ones could be okay.
Do you have a list of the labs that you can send your specimens to? Because
Yeah, at the moment it's 0, so it's a very short list.
well, there's if you go on the web, like what's his name, Michael Krull was studying DNA of morels.
Yeah.
So I sent some morel specimens off to him, and it was like dropping them into a black hole. And I was just wondering, is there any?
So he doesn't actually do the DNA sequencing.
No, I don't. He doesn't do them.
when
He's
did
sort
you
of like
when did you send that?
Pardon me?
He finished that project, so when was it recent?
No, this would be like 6 years ago.
Oh, okay.
That's why I'd like to know, because some of us don't.
And I have to say our lab is something like a black hole too, mostly because we're so backlogged on material, and there's other projects that we're sort of funded to do, which this kind of thing we're not. And so, it usually it usually sits around for quite a while. And in our lab and many others, I think what the expense ends up being is not the reagents so much as the manpower to do it. And usually the people that end up doing sequencing for forays like this are undergraduates, and then it takes maybe a month and a half to get them trained well, and then some of them will only last for another month after that. So you don't get a lot out of them, and even they want some kind of a project that they can wrap up into a paper at the end, because that's what's going to get them into grad school these days. Almost to get into grad school, you need papers. So they'll, instead of looking through everything in a foray, they'll go, well, I'll look at just the Pluteus, or I'll look at just the Suillus, or I'll look at just the Russulas, or something, some project that might come out of it.
So if you had, because I've got what I think would be a great BSc thesis or master's thesis thing for doing DNA and tricholomas. So would I write to universities that have mycology departments and say, hey, I got a chance to give a bunch of specimens to you, and then you strike up how would you, I just like to know.
Actually, Doma, let me just comment. One of the things that we're trying to do as a mycological society, one of the places we've talked to Department of Agriculture, and actually here in Lethbridge, there's a fellow, a mycologist, who would potentially take all the specimens we give him, but what he would like for us to do is to sponsor a student, a summer student. So kind of like what Tom was saying, get a summer student, train him up, and then the student does as many DNA samples as we can send them. So typically what we'd end up hopefully doing in the long run, we were kind of hoping, we've been working on some lottery fund grants, and it's not going really well. So out of those, they really like to give students, scholarships. So those are kind of things. So one of the things that the Mycological Society will probably address in the near future is to create a scholarship be able to send a student to a place like this, and then they'll take care of all of that. And then what we do is we just send them all of our DNA sampling from places, like 4As, and stuff that anybody might send in. Like whether you're sending stuff in from the north, or or people are finding stuff, and then have it processed. So I think that might be our doable way of getting around it. But we need to raise some money.
I think in the long run, what we really want is a central sequencing facility that just does it for a set cost and posts the sequences immediately for anyone that's interested in them to look at. So, like when you send it off, the net result is a sequence posted on the web, connected to all the information that's associated with the voucher from which it came, and then anyone could then look at it and figure out what's going on with it.
Yeah. And for us, actually, sorry for interrupting you. So if we have 200 sequences that come from a 4A like this, and we're talking at, like what Tom was saying, $5 a piece, well, that's $1,000, and there's 60 of us here. Maybe the 4A cost goes up $16, and we fund it that way. But there's all kinds of ways of doing it.
It's actually about $1 a little bit a piece because you've got 4 of them on each card, right?
Well, no, it's still $1 per sequence. It's not. So the card is $1 a piece, even though you pay $4 for the card. And then there's the actual processing, and that's about $5. So there's ways around it, and we're contemplating.
So would we do that so that if we want to see if we've got a certain mushroom, we can send the DNA in?
Absolutely. Absolutely. That's the point.
What was the comment?
Rose was saying more or less that I've got a mushroom that I can't identify, and I can send it in and find out what the hell this thing is. And it's going to cost me however many dollars. That's what she basically says. So in other words, you've got something that's growing in your backyard that you can't deal with. You can do this process and describe it, take a photograph of it, send it to us. We can process it, and at the end of the day, you get your name of what the mushroom is.
Yeah.
Then you can look it up and see, yeah, it's native. Right?
What would be great is like if all the data from a FOIA like this just ended up on like a Google Docs site where anybody could access it.
Yeah.
And then when the sequence comes, it goes right up there on another line for that specimen.
Yeah.
So this is going to put an end to changes.
No.
No, it's actually driving them in some way. No,
They're named improvements. The other benefit is if you put sequences online, the GenBank is the name of the website where people post their sequences that they've. I think they're all required now if they publish paper using sequences. They put the sequences on that website. But if the 4A generates 200 sequences and gets them in GenBank and they're linked to the specimens and the information as to what we thought they were and all that, people that generate their own sequences will find that their thing matches up with our thing, and papers that figure out new species or distributions of things will put your sites on the map. Right now, Alberta's a blank distribution map. Alberta is a blank, right?
There's actually a bunch of mushrooms.
It's changing, but it's changing, but historically there's not been a lot of stuff,
Right.
Because we have, what do we have, 2 mycologists? Here's one, and where's there you are. Do we have more than 2 mycologists in the province? If we do, they're hiding.
I would add one thing to Pat's talk, and that's when he showed the Mushroom Observer. How many people have used the Mushroom Observer? A few of you. explore that site. It is an amazing site, and I would encourage you all to post things there, because what it does is it lets everybody else know right away what the kinds of things you're seeing, and you interact with people all over the world. Some of which maybe aren't positive interactions, but it's
There's discussions.
There's discussions. There's long discussions.
This is fun on Facebook.
But it's
It's better than Facebook.
it's better than Facebook. And,
Oh, it has to be because it's fun on Facebook.
It's got a weighted voting
like when you want to look at what other people think something looks like, you know, if you want to look up who had the hypotheges today, the hygrophorus hypotheges. Somebody had one that we thought it was. So you can go on there and type that in. You'll find like 50 images of it from all over creation. I mean, it's amazing.
And then it makes you wonder. It makes you wonder if what you got is what you think you got.
Right. But
It also tells you what's in your neighborhood.
it tells you what's in your neighborhood, and it gives you an idea how other people, what their concept of it is.
Right. Yeah.
And you won't see all great pictures. You'll see all varieties of pictures. So you'll get a lot of different views of that same fungus.
So what do you know about the DNA? Do you understand all of it and what it tells you about each thing? Like, or do you know that which parts tell you what color it is? Like, do you know those kind of things?
No. Someday maybe. Mostly what we're doing here is we're just taking little bits of DNA that we know something about, sequencing those and using that to infer relationships and species borders. And it's another whole talk on the imperfections of that. It's not a perfect system, but it's a really, really useful tool. Technology is changing so fast that whole genome sequences are now being done for lots of things, and the potential there is that we could look and go, okay, this is this gene is the color that makes the red vermilion streaks in Suillus americanus or something, but we're a long way from that. But I think the potential is there that someday you could actually get more out of the sequence than we currently get.
You used the word earlier on, something to do with collections and NAMA, and it was something like sanctions, or what is the word you use?
Vouchers?
Vouchers, yeah. What is that?
It's a specimen that documents a name.
Or an observation. So like when you have a whole list of collections that you made here, or observations that you made here, if the only thing you have is that name, then all we know is that somebody thought that that's what you saw. If it is anchored to a specimen, a voucher, then someone else can look at that and go, okay, do I believe that that's what this was? And especially as the names change over time.
So who creates this voucher? Is it a number?
You are here. This is when you dry these things and have this specimen that goes into a herbarium somewhere, that is a vouching specimen.
So if you look over there on the stuff that is on the display, the vouchers have a line across the card to make it obvious that that specimen has to go in the dryer at some point to be saved for whoever it is that wants, or that's saved for somebody. Yeah.
So if you get two DNAs that kind of match, but you have two different things, how are you going to know which one is really the right one?
Oh, and that's actually a common occurrence because the database for DNA is awful. So because there's a lot of stuff that gets put into it that's wrong. There's names that haven't been updated. There's all kinds of things. So it gives you more information, but it's not automatic yet. And there's there's a lot of work being put into the database now to try to improve it.
Basically improving the vouchers and information.
To improve the vouchers
sequences
and
based.
also to annotate the database so that people actually look through the sequences and go, okay, this cloud of sequences that has 3 different names is really this name. So that has not been done yet, but it's just started to get worked on. And it's really important because in the world of ecology, fungal ecology, everything is sequenced now. That when we look at fungal communities, it's all sequence based. You know, you just take the soil, grind it up, and sequence everything, and then you can say, okay, well, there's these 6 trichomas in there, and there's this pseudomonas and all these penicillia and blah blah blah, and the whole big laundry list. But that list is only as good as the database, and so it's critical now that these databases get corrected and updated. And the best way to do that is to have new collections come in that have vouchers associated with them, because a lot of what we're finding is that when we're comparing sequences now across, especially North America and Europe, is that we have different stuff. We've been calling it the same thing, kind of the Lactaris deliciosus example that's here. We have things that look really, really similar and that we've been applying European names to, but it turns out it's not the European species. And so when we use data from Europe and try to match it, we go, well, it's close, but that's not quite right. And then we've got these other sequences from Alberta that match, but we don't have a name for it. So that alerts us that, okay, there's these things that are parading under European names, but are really North American endemic species that need to be described.
Or they're in the east and you have a different thing in the west.
Yeah. Very common. I'll show a couple examples of this tomorrow night when I talk about the microflora.
Excellent. One of the things I'm going to point out, I think that when are we going to start doing hammering out some extractions? After Paul's talk tonight?
Yeah, we could do that.
We can start with that. So what I'd like to do is, we're going to do some of those, and I would really encourage, and I'm probably even going to, if I don't see somebody over there, I'm going to elbow you and send you over that way. Because I'd like us to do that with Tom, with some of our stuff, and just learn it. And also what I'd like to do is, we're going to have some mushrooms here that we're going to struggle with in identifying. And we've already decided that what we're going to do is describe them. And so Patrick's going to work a little bit on that. And what I'd like to do is, I'd like to grab some of you to actually sit down with him and start going through some of these mushrooms and describing them and getting the notes on the fresh specimen, and then it'll get smashed with the hammer for the DNA. Well, actually, it'll get photographed and then smashed and then dried. And then, because it might be something we don't have a name for. And I think that's something that I'd like us guys to do. And I see lots of smiling faces who'd love to do that.
But it seems like it would just be in the circuit where somebody's going to a dryer that's smashed before closing the dryer.
So let's kind of get actively involved in this citizen and in citizen science and let's own it. And because at times we're not going to have these guys around, and then we know what to do, we know what it takes, when we find something really unique and different, we can deal with it. Absolutely, we're pioneering this. We here are pioneering this for North America because we're going to try and figure out how it works here.
Foray with smashing hammers.
That's right, smashing hammer foray, and then from there, hopefully it does the tour across all of North America. Melanie,
So our dinner has been ready for a while. So we should maybe push these specimens in the center and eat around it, or how do we?
well, actually, whoever specimens these are, you guys know who you are. Take them and put them on the table. And,